Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk a little bit about protein digestion and absorption. Just as a reminder, we are still talking about this state, which is after a meal or postprandial. In the last video, we talked about the synthesis of the non-essential amino acids. In today's video, we shall look at the digestion of proteins in the GI tract and then how we absorb the essential as well as the non-essential amino acids into our bloodstream. So we'll begin our discussion with protein digestion into chemicals that we can absorb, and that would mean the amino acids as well as some small peptides such as dipeptides and tripeptides. It's not only proteins that are in your food, the dietary proteins that we have to take care of, but we also produce a lot of endogenous proteins that will be digested. So the examples of this would be the mucins that are produced by the GI tract, the epithelial cells that line the GI tract. They are sloughed off very often, and they have to be, all the proteins in these cells have to be digested, and a lot of digestive enzymes are also proteins that are produced by the GI tract that undergoes digestion. So nothing happens much in the mouth. There is mechanical breakdown by the process of chewing, and then the food is mixed with the saliva and then swallowed. The food then enters the stomach, and there are two important processes we'll talk about here. One would be the secretion of hydrochloric acid by the parietal cells, and the other would be the secretion of the enzyme pepsinogen by chief cells. So let's first look at hydrochloric acid production by parietal cells. What is the biochemistry of this process? So here is a parietal cell. This is the stomach cavity, and this is the bloodstream here, a blood vessel, a capillary. So how do we produce hydrochloric acid? So let's look at the steps by which the parietal cell secretes this hydrochloric acid that provides the stomach cavity a pH around 1.5, so very strongly acidic. So we'll start the story here in the capillary, in the bloodstream. Carbon dioxide, which is non-polar, can easily cross through membranes. So it will cross through the endothelial cell that line the capillaries, and then it will enter the connective tissue, and from there it will cross the basement membrane and then pass through the plasma membrane of the parietal cell, and it will now enter the cytoplasm of the parietal cell. Since we are removing carbon dioxide from our bloodstream, the pH of the portal venous blood, the pH here, will slightly increase, and this is called the alkaline tide. So after a meal, the pH in your bloodstream, in your portal venous blood, increases, makes it slightly alkaline, and that is called the alkaline tide. Now once in the cytoplasm, the enzyme carbonic anhydrase that we talked about in a previous video, a very important enzyme. So this enzyme catalyzes the reaction of carbon dioxide plus water. So CO2 will react with water to give you carbonic acid, H2CO3. Now carbonic acid is unstable and it will break down to give you protons and bicarbonate. So we get protons and bicarbonate produced in the cytoplasm of the parietal cell. These protons that are produced will now be exported out into the stomach cavity in exchange for potassium that will enter the cytoplasm. And this exchange is carried out by pumps. These pumps are called the proton potassium ATPase pumps, and these are found on the apical membrane of the parietal cell. Since these are pumps, they would need a lot of energy, so they would use quite a bit of ATP to pump protons from the cytoplasm into the stomach cavity. So since this proton pumping is done with the help of bringing in potassium, so we need to have enough potassium in the stomach cavity. So the next question would be, where does this potassium come from that is required by these pumps? So we have the basolateral sodium potassium ATPase pumps. So here we have the sodium potassium pumps, and as you know, this will export sodium out of the cell and in exchange for potassium, which is imported into the cell. So this way potassium comes into the cytoplasm of the parietal cell. Now this potassium in the cytoplasm is then sent out into the stomach cavity, and this happens through apical non-gated potassium channels. These channels are open, and the potassium this potassium here basically leaks out of this channel and enters the stomach cavity, and this potassium can then be used by these pumps here that pump the protons out. So while the protons that are formed from this carbonic anhydrase reaction, while these protons are sent into the stomach cavity, the bicarbonate, the other product, is exported through the basolateral membrane into the bloodstream. So this bicarbonate is sent out by these proteins, transport proteins called basolateral ion exchanges. They will export bicarbonate while at the same time bringing in to the cell chloride ions. So this is an antiporter. It's a bicarbonate chloride antiporter. So chloride from the bloodstream is brought into the cells. Bicarbonate in the cytoplasm is sent out into the bloodstream. Now nearby to the parietal cells are epithelial cells of the stomach, and so this bicarbonate in the bloodstream will travel through the bloodstream, and they will reach the epithelial cells that are also present in the stomach. The epithelial cells will now use this transport protein called bicarbonate sodium symporter. So basically it will bring in bicarbonate as well as sodium into the cytoplasm of the epithelial cell. This bicarbonate that is brought into the cytoplasm of the epithelial cell then is exported into the stomach cavity by a bicarbonate chloride antiporter. So this transport protein will send bicarbonate out in exchange for chloride, which comes into the cytoplasm from the cavity. So on the surface of the stomach we have this lining, this mucus lining. Mucus comes from mucus cells that are present in the stomach. So right here on the surface we have mucus, and now this bicarbonate mixes with the mucus and basically makes this mucus lining on the surface of the stomach, and this provides the stomach lining a protective alkaline mucus blanket. So that's how the stomach protects itself against the corrosive nature of this hydrochloric acid that it is producing and secreting. So we talked about the proton part of HCl. Now what about the chloride part of HCl? So where does this chloride come from? And the answer is the chloride comes from the bloodstream. As we mentioned earlier, the chloride bicarbonate antiporter will bring this chloride from the bloodstream into the cytoplasm, and from the cytoplasm the chloride will leak through the apical part of the membrane of the parietal cell membrane, and it will leak through via these non-gated chloride channels. Okay, so the chloride gets out through the non-gated chloride channels and will appear in the stomach cavity. So we have the H+, we have the chloride minus, and this will give us hydrochloric acid. Now the parietal cells have a very large, extensive network of mitochondria, have many abundant mitochondria, and that makes sense because of these pumps here that require a lot of energy. And recall that most of the ATP production happens in the mitochondria. So now let's see how the presence of hydrochloric acid helps in the digestion of proteins. So while the parietal cells are producing hydrochloric acid, the chief cells in the stomach produce an inactive enzyme called pepsinogen. So these proteins that need to be digested, whether they come from the diet or whether they are endogenous, in the presence of the strong acid pH 1.5, remember we talked about this under proteins in very early videos, all the amine groups will become protonated, all the carboxyl groups will become protonated. When carboxyls are protonated, they have no charge, but when amine groups are protonated, they are positively charged. So wherever there are amine groups, there will be positive charges. So this protein will be filled with positive charges. As you know, positive and positive repels, and that causes the protein to unfold. In fact, this is one of the strategies we use even in the labs to unfold proteins, and that is by putting the protein in a strong acidic environment. So in this case, the acidic environment is provided by hydrochloric acid. So the acidic environment also will make the inactive pepsinogen that is coming from your chief cells, secreted by chief cells into the stomach cavity, and this pepsinogen becomes a very active enzyme called pepsin, and now wherever in the unfolded protein chain, wherever there are aromatic groups or acidic groups, so wherever there are aromatic or acidic groups, this pepsin will break the peptide bond and produce the smaller peptides, which will be much more difficult to fold because they are much more smaller. So what we get is unfolded protein fragments. So in many of the proteins that we are going to digest, this is what we might end up with, small protein fragments that are unfolded. So these products of pepsin digestion as well as other proteins will now enter the intestine, the duodenum, And let's see what happens in the duodenum. So when the food enters the duodenum, these cells, these endocrine cells present in the duodenum, they are stimulated to secrete cholecystokinin, CCK. And this CCK, together with the acetylcholine that comes from the vagus nerve, it will stimulate the pancreas, the pancreatic acinar cells. So here you have these acinar cells in the pancreas, and these cells will be stimulated to secrete zymogens. This process happens by exocytosis. So these cells via exocytosis will release zymogen granules. These granules contain a lot of inactive enzymes such as chymotrypsinogen, trypsinogen, proelastase, and many other inactive enzymes. So these zymogens, together with a lot of bicarbonate, this bicarbonate is made from the ductal cells in the pancreas. So together these inactive enzymes and bicarbonate will be secreted from the pancreas. From the pancreas here, it will enter the pancreatic duct, and from the pancreatic duct, it will enter the duodenum. So now the pancreatic juice is mixed with the food in the duodenum. Now there are enzymes on the intestinal cells. These enzymes are called enteropeptidase or enterokinase. And these enzymes basically convert the inactive form, trypsinogen, into the active enzyme trypsin. This active trypsin will now, in the cavity, it will now convert the inactive chymotrypsinogen into enzymatically active chymotrypsin. Similarly, other inactive enzymes such as proelastase will be converted into the active elastase form. So all of these active enzymes that are produced now in the intestinal cavity, what we call pancreatic proteases, trypsin, chymotrypsin, carboxypeptidase, elastase, and several more. These active enzymes will now break down all the proteins as well as the pepsin-derived protein fragments that are found that are present in the small intestinal cavity. So all of these proteins will be broken down into dipeptides as well as tripeptides and some oligopeptides. There are enzymes called peptidases. These are present in the brush border of the intestinal cells. So these enzymes are peptidases, and these enzymes will break down the oligopeptides into dipeptides, tripeptides, and amino acids. So at the end of all this digestion, we now have the amino acids, which could be either the neutral ones, the anionic ones, the acidic, as well as the basic or the cationic ones. And we have some dipeptides as well as tripeptides. And now we are ready for the next step, and that is the absorption of these chemicals into the intestinal cells. Let's first look at how the neutral amino acids are absorbed. There are several different proteins that are involved in the absorption of amino acids. So we'll talk about some of the major ones that have been identified. So for the neutral amino acids, we have on the apical side, that is facing the cavity, we have these proteins called sodium-dependent broad neutral amino acid transporters, B0AT1. It's basically, these proteins are symporters, so not only do they bring in the neutral amino acids, they also bring in sodium from the cavity into the intestinal cells. These transporters prefer the branched-chain amino acids and methionine, although they transport all kinds of neutral amino acids. So once in the cytoplasm, these neutral amino acids are then sent out into the bloodstream. So we have these basolateral transport proteins. And examples of these basolateral transport proteins for the neutral amino acids include the large neutral amino acid transporters LAT2, LAT4, as well as the system T amino acid transporter 1 or TAT1. The neutral amino acids will then come out into the connective tissue and then will enter the capillaries. For the anionic amino acids, that means the acidic amino acids, the apical protein that brings in the acidic amino acid is called excitatory amino acid transporter 3 or EAAT3. This is an antiporter. It's a complex protein that brings in sodium as well as the anionic amino acids such as glutamic acid and aspartic acid. So it brings these things in at the same time exporting potassium out of the intestinal cell. So once the acidic amino acids, glutamic acid and aspartic acid enters the cytoplasm of the intestinal cells, they're basically metabolized in the intestinal cells, in the enterocytes. So there are no identified proteins or basolateral membrane proteins that have been identified that actually export these anionic amino acids out of the cell. Now the cationic amino acids or the basic amino acids, on the apical membrane we have these transport proteins called sodium independent blastocyst neutral and cationic amino acid transporter B0 plus AT. And these proteins are antiporters, so they basically bring in the basic amino acids into the cell, arginine and lysine, while at the same time exporting the neutral amino acids. So neutral amino acids are exported out of the cell, cationic amino acids are brought into the cell. So once these cationic amino acids are in the intestinal cell, then on the basolateral membrane, we have these proteins called cationic and large neutral amino acid transporter 1 or Y plus LAT1. And this protein basically is an antiporter, so it will export the basic amino acids, arginine and lysine, out of the intestinal cell, while at the same time bringing in the neutral amino acids. It will import the neutral amino acids into the cell. And once again, these amino acids will enter the bloodstream and go to the portal venous system. There are also proteins that absorb or transport dipeptides and tripeptides. So on the apical part here, we have the PEP-T1 proton peptide co-transporter. So this brings in not only the dip and tripeptides, but it also brings in protons. And once the peptides, the di or tripeptides are in the cytoplasm of the intestinal cell, these peptides are then broken down into single amino acids, and then these amino acids will use the transport proteins present on the basolateral membrane, depending on what type of amino acid it is, whether it's neutral or it is cationic. So that's how the amino acids are transported from the intestinal cavity into the intestinal cells and then eventually into the bloodstream. Now, what about intact proteins? Do we absorb intact proteins? So the latest research shows that adults probably do not absorb intact proteins, or we absorb very, very negligible amounts, if any. However, important to note that neonates, the first few days after birth, they do absorb intact proteins, probably by the process of pinocytosis. And this is important because they do bring into the bloodstream immunoglobulins that is found in colostrum, the milk from the mother, and these immunoglobulins will provide them with passive immunity. A few words about the transport of amino acids in the bloodstream. If you look at the plasma concentration of amino acids, it's usually less than 1 millimolar. That's we're talking about less than 1,000 micromole per liter. For example, one of the most hydrophobic amino acids, phenylalanine, its concentration in the bloodstream is only around less than 0.1 millimolar. The highest concentration in the bloodstream is glutamine. They can have a concentration as high as 0.8 millimolar, alanine 0.5 millimolar. Now, the solubility of amino acids in water, for example, we take again this hydrophobic amino acid phenylalanine, is quite high, 150 millimolar. So clearly you can see that these amino acids are found at concentrations much lower than their water solubility. That means most of these amino acids can easily dissolve in the plasma water, and that's how they will travel in the bloodstream. Now, the cationic amino acids such as your arginine and lysine, because they have a net positive charge, it is possible that they might bind proteins such as albumin in your bloodstream, which have a net negative charge. So before we end today, a few words about lab tests. We can measure amino acids, the different types of amino acids in the bloodstream. So we do that test. That test is not done routinely. It is ordered for special cases. However, routine protein tests include albumin. We do measure the concentration of albumin in the bloodstream. We do measure total proteins. And from these two, we can find out the albumin globulin ratio. And that's basically calculated by dividing albumin by the total protein minus albumin to give you the AG ratio. So we'll talk about these tests later under clinical cases. So that's all for this video. Once again, thank you very much for watching. Have a happy and a healthy day.